Is it audible now?
Yeah, it's audible, Froze. It's audible.
Hello? Okay. So, good morning, all of you. I welcome all the members and the participants who are virtual now, and let us start the inaugural program. We will start the program with a formal prayer, and now I call upon Muhammad Ubada to recite the Kirat.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم قد أفلح من زكاها وقد خاب من دساها كذبت ثمود بطغواها إذ انبعث أشقاها فقال لهم رسول الله ناقة الله وسقياها فكذبوه فعقروها فدمدم عليهم ربهم بذنبهم فسواها ولا يخاف عقباها صدق الله العظيم.
Now I call upon Dr. R. Rafi Muhammad, the head of the Department of Zoology, to welcome the virtual gathering.
Assalamu alaikum. Good morning, everybody. First of all, I thank Almighty Allah for giving me the opportunity to stand before you. I deem it a privilege to welcome you all for the International Virtual Conference on Modern Zoology, Known and Unknown. I deem it an honor to welcome our dynamic correspondent, Hali Janab S.J. Abrar Ahmad Sahib, to this program. Welcome you, sir. I feel extremely happy to welcome our beloved principal, who always encourages us to conduct this kind of programs, which all its support. I welcome you, sir. I also extend a warm welcome to Dr. Ibrahim Sharif, sir, Vice President of College Association. Welcome you, sir. I take pleasure in welcome our esteemed chief guest, Dr. M. Mufizur Rahman, Research Scientist, PH Cologne University, Applied Science, Germany. Dr. S. Manoharan, Researchers, King Fahd University, Saudi Arabia. Dr. Ganapati Venkatachalam, Professor, Anna University, India. Dr. P.R. Jayachandran, Researchers, Cochin University of Science and Technology, Cochin, India. Dr. B. Gunalan, Assistant Professor, Center of Advanced Study in Marine Biology. To our college virtual conference, who will share their knowledge and vast experiences. Once again, I welcome you, sir. My hearty welcome to organizing secretary and coordinators and colleagues and staff members of our college and other colleges. I welcome the participants and the students various parts of India and other countries. This conference program welcome you. Thank you.
Now I request Dr. K. Jahir Hussain, Assistant Professor of Zoology and the Vice President of Zoology Association, to share about the conference. Over to Dr. Jahir.
Thank you, Dr. Feroz. Very good morning to all the participants and dignitaries. I am glad to introduce the importance and the theme of the webinar. Before elaborating about this extraordinary meeting via web, I would like to say a few words about the department and the inception of this webinar. The C. Abdul Hakim College, managed by Mel Visharam Muslim Education Society, was established in 1919. It is located at Mel Visharam, Ranipet district of Tamil Nadu, approximately about 120 kilometers from Chennai. The college has been reaccredited by the NAC and conferred the autonomy by UGC. It offers programs from UG to PhD. The Department of Zoology of this college is addressed as a nerve center of research activities and innovation. The department was started in the year 1966 and PG was offered during 1981. Subsequently, the research programs were started in 1986. More than 350 research publications have been published by the staff of this department, with total impact factor of 500, and the total citation during the last 5 years is about 6,500, which is a dream of any university department and any research institutions. The staff members have collaborated with many national and international institutions and awarded with more than 20 crores of rupees from the funding agencies. The Zoology Association was started in our department in the year 1996 with the aim to enlighten the young minds of CAHC with the current trends through the lectures of eminent personalities. Similarly, the FIMZ, Frontiers in Modern Zoology, is a platform that familiarize the modern aspects of biology to our students and fellow biologists through such programs. This webinar is a brainchild of Dr. Ansari, who has taken meticulous effort to assemble the resource persons. We received the overwhelming response with about 600 registration from India and abroad. Similarly, the center point of this webinar is rapid population growth and technological innovations combined with our lack of understanding about how the natural systems of which we are part of work have created a mess, and our sincere effort to reclaim our environment. With specific thrust areas like climate change and global warming, environmental pollution and control, biodiversity and conservation, ecotoxicology, environmental impact assessment, environmental ethics, sustainability, applied zoology, and molecular biology. During this webinar, we are going to listen from many eminent scientists who are working on these thrust areas. I congratulate and welcome the participants and each and everyone associated with this webinar through net. Thank you.
Thank you, Dr. Zahir, for the excellent introduction about the conference. And now I request our principal, Dr. S.A. Sajid, to give the presidential address.
Assalamu alaikum wa rahmatullahi wa barakatuhu. Happy morning. Most respected head, Department of Zoology, the conference convener, the organization secretary, the conference coordinators, the organizing team, resource persons, and keynote speakers, dear participants. I welcome you all for this international virtual conference on modern zoology, known and unknown. At the outset, I congratulate the Department of Zoology for our college for having taken the keen interest to organize this type of program. It is a great service to the society at large. The actually the people like you, those who are involved in biological sciences, how to take the responsibility to control everything which is got going to take which is all taking place in the world. So this is a webinar. Definitely this conference will be useful to all the participants to get an idea and to involve themselves in active research so that the maximum outcome will be easily brought out to the society, so to come over with all these difficulties. Many areas of study in the biological sciences are known in the modern world, and many areas are unknown. And to comprehend this, researchers must cross the barriers that have historically separated the various schools of science. The role of biologists in society, as well as their moral and ethical responsibilities in the discovery and development of new ideas, led to a review of individual, social, and scientific value systems in the 20th and 21st centuries. The 20th and 21st centuries also saw significant developments in areas of biology concerned with ecosystems, the environment, and conservation. Climate change, growing significance, and impact on ecosystems drove improvements in ecology as well as the establishment of specialties such as conservation biology and conservation genetics. Other issues confronting biologists included the hunt for strategies to reduce environmental contamination while not interfering with efforts to improve humankind's quality of life. The problem of excess human population contributed to the pollution problem. Increased worldwide human population created higher demands on the land, particularly in the field of food production, and necessitated an increase in modern industry operations, the waste products of which contributed to air pollution, water, and soil. Biologists collaborated with social scientists and other elements of society to discover the needs for maintaining an healthy and productive world in order to develop solutions to global warming, pollution, and other environmental concerns. Because while many of humanity's current and future issues appear to be primarily social, political, or economical in origin, they have biological repercussions that threaten the very existence of life itself. Based on the scenario described above, this virtual conference would aid in bringing scholars together and the researchers, and they will going to suggest the known and discovered aspects of modern zoology. We are grateful to the keynote speakers and the speakers who are going to give special talk on this conference, particularly Dr. M. Mafizur Rahman from Germany, Dr. S. Manogaran from Saudi Arabia, Dr. Ganapati Venkata from Anna University, India, Professor Dr. P.R. Jayachandran, research scholars from Cochin University, and Dr. B. Gunalan, the researcher and assistant professor from Annamalai University, India. We are grateful to you all, sirs, for having accepted our invitation and who are going to deliver the very important lectures on the topic. I congratulate all the participants, researchers across the world for having joined with us in this virtual conference. I definitely am very grateful to you all for having joined with us. We wish this conference should be a successful one to bring the solution to all these problems which are going to be discussed in these 2 days. I thank the organizers for having given me this opportunity to say a few words about this virtual conference. Thanks.
Thank you so much for giving the valuable address. And now we go into the technical session 1. Now it's over to Dr. Khader Shah to invite the speaker 1.
Hello. Hello. Dr. Jayachandran, are you there?
Yes, I am here.
Okay, thank you.
Okay.
Good morning to all. It gives me an immense pleasure in introducing Dr. Jayachandran PR, who is currently serving as a postdoctoral fellow in the Department of Marine Biology, Microbiology, and Biochemistry from the School of Marine Sciences, Cochin University of Science and Technology, Kochi, India. A doctoral holder in the field of marine biology and a net qualified candidate. His broad research area compiles of marine biology, benthic ecology, marine ecotoxicology, polar biology, Arctic benthas, environmental monitoring, bioinformation ecology, biogeochemistry, and then amoeboid protists. To his employment credits, he was a junior research fellow and senior research fellow in state and central government projects from the Government of India. He is having 12 years of research and teaching experience in various research as well as academic projects, academic firms. And then regarding his administrative skills, he was a project scientist too of MOS, NCCR organizing international training and workshops as an organizing member and associate organizing secretary. To his editorial credits, he is a subject editor in the Journal One Ecosystem, Pensoft publishes SCI journal reviews, and then he has experienced 3 Indian Arctic expeditions in the comprehensive years 2016, 17, and 18 to study the ecology and diversity of benthic amoeboid protists, diversity of fjords of Svalbard, EAA studies in coastal waters of Kerala and Lakshadweep, water quality monitoring including microbial contaminations. He is having field cruise experiences in open seas, Arctic fjords, estuaries, mangroves, seagrass beds, and coral reefs. To his credits received, he had received a distinguished research award of QSAT 2019 and a PADI certified open water scuba diver. He published more than 30 full-length articles in SCI referred journals, 6 books and training manuals, 14 book chapters and proceedings in various seminars as well as conferences. He published 21 abstracts. He had given 8 invited talks and presentations and 2 popular articles. To his credit, he is having total citation of 262 with a H index of 10, I-10 index of 10. With a total cumulative impact of 44.71. Thanks for the opportunity given. Send your presentation, sir, please.
Okay. I thank you, coordinators of the conference, for giving me an opportunity. And can you hear?
Yeah, it's audible, sir.
Yes, yes, sir. I wish you all good day. And can I share my screen?
Yes, sir, you can share your
Okay.
screen.
Okay. 1 minute.
ஏன் இவன் இதுல வர உமர் அலி எதுக்கு இதுல பையன் தான் இதுல தானே இருக்கான். One sec.
സമയം ഇത് ഇതാക്കണം അങ്ങനെ stop sharing. 1 minute, I'm sharing. Is it visible now?
Yes, sir, it's visible.
Yes.
Yeah, it's visible.
Okay. Now we are going to discuss about the biological carbon cycling in ocean and its importance. It's a very important topic in our day-to-day life. And United Nations estimates today more than 40% of the world population living within the 100 kilometers of our coasts. And many coastal regions are subject to intense land use and in turns also intense degradation. And eutrophication of coastal is due to the nutrient from the agriculture sources is a major problem we are facing, and it results in a strong algal growth, and when it gets decay results in oxygen depletion. The sea is of a fundamental importance to humankind due to the many ecosystem services it provides. And marine experts divided this ecosystem service into four classes as professional services and supporting services and regulating services, and also a cultural services they are providing to us. The professional services includes the production of marine fishes and for our human consumption. And the cultural service it comprises the tourism and other traditional concern, the connectedness to the sea, like boat building and all. And supporting services includes the first and foremost mainly the primary productivity of oceans, that is the generation of biomass by the phytoplanktons by means of the photosynthesis. Finally, the regulating services they provide are taken by scientists into the include fundamental biological, chemical, and physical process in the oceans, and such as the nitrogen and carbon cycles, as well as the ocean's current, which affect the terrestrial climate. The elimination of the marine pollutants is another regulating services offered by marine ecosystems. Many of these services are now under the threat from human overexploitation, environmental pollution, or greenhouse gas emissions. The carbon dioxide emission in particular pose a global threat to oceans today. First, these emissions are causing the oceans to warm as a result of greenhouse effects. Secondly, a significant proportion of the atmospheric carbon dioxide become dissolved in the seawater and slowly acidifying the oceans. And given the climate crisis, it's urgent to reduce the anthropogenic carbon dioxide emissions and explore climate geoengineering opportunities for mitigate these effects. And carbon compounds regulate their temperature, make up food that sustain us, and provide energy that fuels our global economy. The most of the Earth's carbon is stored as rocks and sediments, and the rest of it is located in our oceans and atmosphere, also in living organisms. These are the reservoir through which carbon cycles. How carbon and carbon storage and exchanges happens. As we know, carbon move from one storage reservoir to another through a variety of mechanisms, such as in the food chain. The plants move this carbon from the atmosphere into the biosphere through the photosynthesis. They use the energy from the sun to chemically combine the carbon dioxide with the hydrogen and the oxygen from the water to create the sugar molecules. These animals that eat this plant digest the sugar molecules to get the energy for their bodies. And the respiration, excretion, and the decomposition process release the carbon back into our atmosphere or to the soil, continuing these cycles. Changes to carbon cyclings are happening now. The human activities have a tremendous impact on the carbon cycles. Fossil fuels, changing the land use, and the using of limestones to make the concrete are transfer significant quantities of the carbon to our atmosphere. As a result, the amount of carbon dioxide in our atmosphere is rapidly rising. It is already greater than at any time in last 3.6 million years. The oceans absorb much of the carbon dioxide that is released from burning of fossil fuels now. This extra carbon dioxide is lowering ocean's pH. That is very important through the process called the ocean acidification. Due to the ocean acidification infers the ability of the organisms, including the corals, crabs, snails, to build their shells and skeleton. This is very important aspects we need to study more. An accurate understanding of the process and the mechanism of the carbon cycles and its interaction with the human driven climate change is fundamentally important. The aquatic microbiology section made an important contribution to new knowledge on these aspects and insight to this research field, and including focusing investigation on the marine primary production, the organic matter biodegradation, and the biotransformation, and also the microbial process response to the natural and anthropogenic environment gradients and stresses. Oceans play a critical role in the global carbon cycling and may provide a solution for a climate change mitigation also. How oceans play a critical role? Our oceans cover around 71% of our planets, and it serve as a major repository for anthropogenic carbon dioxide. And oceans play a critical role in carbon storage, as it's hold about 50 times of more than carbon than the atmosphere, and because it's called 50% of the atmospheric carbon dioxide in the ocean as a dissolved organic matter. The physical factors and biological process, biotic process determine the fate of the carbon in our ocean. The major reserve of carbon occur as a carbonate minerals in sedimentary rocks in the earth crust, and also in inorganic compounds such as the corals, oil, and natural gases. Turnover of this carbon by natural geological process is very slow. However, in last few hundred years, the human activities have accelerated this flux of the carbon to the biosphere because of the use of the fossil fuels and change in the land use pattern. Atmosphere currently contain around 858 gigaton of carbon, and the concentration has risen rapidly since the pre-industrial time to around now the present level is around 405 ppm, and it is highest level in the past 8 lakh years. That is important. Oceans are estimated to contain around 662 gigaton of dissolved carbon, is about the 3/4 of that in our atmosphere. In North Atlantic and the Southern Ocean, the cold and the saline and dense water is a high carbon dioxide. It's contain the high carbon dioxide. It vertically sink into the depth to 2,000 to 4,000 meters, and then it's distributed throughout the ocean basins or ocean bottoms. And mass of water sinking into deep oceans is replaced by the welling process gases to surface elsewhere. As water warms, the carbon dioxide escapes to atmosphere again. And this process of the carbon dioxide cycling in our ocean is also known as the dissolution pump or the solubility pump or physical pump. In addition to purely the physical factors, the biological carbon product pump is responsible for the massive distribution of the organic carbon in our ocean. The fixation of carbon dioxide by the phytoplanktons in upper oceans leads to the primary production by incorporation of carbon into cellular materials. The Prochlorococcus and Synechococcus are the pico-sized cyanobacteria play a fundamental role in the ocean primary productions, being particularly important in warm, nutrient-poor waters like the oceanic gyres. Their potential response to nutrient enrichment is expected to contrasting and to differ from the large phytoplankton species that dominate in coastal waters like diatoms, dinoflagellates. However, the Prochlorococcus species contribute around 20% of oxygen generation to atmosphere global average. These phytoplankton cells may be consumed by zooplankton or fishes. So some of that carbon entered to food chain directly. Significant amount of fixed carbon is released as a dissolved organic carbon from phytoplankton cells by the exudation process, and when it die and also break up either by the natural sense or due to the virus lysis or bacteriophage lysis. The polymeric substances like the transparent exopolymeric particles and cell debris formed in the photic zone of is formed particulate organic matter on the ocean surface and which aggregate together with the calcium carbonate and the silica cells of the protist available in the upper water column to form the large particles of marine snows. These marine snows settle into deep waters by gravity, vertical migration of zooplankton, or by the subduction of water masses. On this passage through the water column, a large fraction of this fixed carbon is required by the microbes and will be returned to the atmosphere. And about 25% of the primary production reaches to our seafloor. There, most of the carbon compounds are broken down by the microbial fermentation or by the respiration process. About only 0.3% of the primary production will be buried into deep sea sediment and over 4 millennia. And this residue of buried carbon is transferred by the increased exposure to heat and pressure form our reservoir of hydrocarbons that we are using now.
Excuse me, sir. Sir,
Okay.
your slides are not moving, sir. They're just still. It's in the first slide itself.
Oh, not moving. Huh?
Yes, sir. The
Okay.
display is
Okay.
just on the first slide. It's not moving down.
Okay. Okay. I'll check. Okay.
Thank you, sir.
It's not moving.
Now it was moving, sir.
Okay, but when full slide, okay, I will do like this.
You have to share the window, sir. If you share the whole screen, might be it will be a problem. If you
Okay.
share the window alone, and you need to keep your presentation open and keep pressing next.
Okay, I will do like this. Sorry.
Yes.
So it's okay. There's some problem with the presentation. Okay. Okay. It's okay.
Thank you, sir.
Okay, the microbial loop, and now we are going to this. The microbial loop, viral strand, and microbial carbon pump components of the carbon cycling systems provide both sink for the fixed carbon as well as a link to the higher trophic levels. About half of the dissolved organic matter resulting from the phytoplankton production passes through the microbial loop, acting as a source of energy, carbon, and other nutrients for the heterotrophic organisms. Results. Can you see now?
No, sir. Your screen is not visible. You're supposed to again share your screen.
Oh, not seeing. Okay.
Yeah.
What happened this time? Enter screen, but share. No, it is not coming. 1 minute. I will use the Windows. Okay, I will do like this. So can you see now? Hello.
Yes, and now it is visible.
Okay, okay. Now we'll do like this. So I'll repeat the microbial loop. The microbial. Sorry for inconveniences. I have not noticed this problem with that. The microbial loop and viral shunt and the microbial carbon pump component of the carbon cycling system provide both a sink for the fixed carbon as well as the link to higher trophic levels. About half of the dissolved organic matter results from phytoplankton production passes through this microbial loop, acting as a source of energy, carbon, and other nutrients for the heterotrophic organisms. This results in the greater retention of dissolved nutrients in the upper oceans, but as mentioned above, the microbial loss of the carbon compound alter the elemental composition of our results in the formation of recalcitrant dissolved organic matter in the water column, which is resistant to biological degradation, which is transported to deep ocean and sequestered for a long period, even for a millennia. Many phytoplanktons are responsible for about half of our global primary production. The primary production can be defined in terms of the total amount of carbon dioxide fixed into the cellular materials during the daytime, which is known as the gross primary production, as we know, and as fraction that remain after the loss of the respiration by the phytoplanktons, which occur both day and night, which is known as the net primary production. Another concept is the net community production has been introduced to encompass the effect of the respiration by the heterotrophic organisms. Both biomass of the plankton and the rate of the gross and net primary productions are affected by a range of factors, which vary greatly over the both long-term and short-term time scales, into the interplay of the hydrographic conditions with the light conditions and nutrient availability. The temperature increases both photosynthetic and respiratory rate, but this effect is complicated in oceans. The quantifying the photosynthetic productivity has been a central question in biological oceanography for more than a century. Various methods are available, and these maybe produce different results depending on the parameter measures and time scale employed. The most common laboratory experiment measures incorporate of the radio-labeled bicarbonate into the organic materials, comparing the effect of incubation in light and dark bottles. We know these processes. The discovery of widespread anoxygenic phototrophic bacteria means that is earlier assumption that based on the use of oxygen evolution need to be reconsidered, because as these bacteria are through to be responsible for about 5% of the primary production by anoxygenic phototrophic bacteria. The photosynthesis rate are measured over time under different environmental conditions, including the light. The batch culture and bottle experiments obviously have inherent limitation, because the nutrients are quickly depleted in the absence of diffusion, and artifacts can be introduced during the deployment. And retrieval of the samples by the contamination or by the alteration of the spectral qualities of light entering to containers. The mesocosm experiments in large natural enclosures containing several cubic meters of the seawater or semi-continuous or chemostat culture in laboratory can overcome some of these problems. Since distribution of the primary production is highly dynamic and influenced by many factors, reliable estimates of the productivity throughout the ocean have only been possible since the advent of 1970s remote sensing process using the spectral scanners of the satellites, which measures ocean's color. The NASA's C5 satellite sensor, which is operated from 1977 to 2010, and its successors operated by the International Space Agency, have produced a vast database on global ocean color and other measurements that can be used to track the regional and temporal variability in the phytoplankton communities, allowing estimates of primary production. In clear water with little suspended materials, many factors influencing the ocean color is the light absorption by the phytoplankton pigments, especially the chlorophyll. Therefore, water appears blue when there are low density of phytoplanktons, and greener when there are high density of plankton. Properties of the chlorophyll and other photosynthetic pigments. It is possible to map surface phytoplankton biomass daily if cloud cover is permitting. Furthermore, the depth of the mixed layer, which can vary fivefold, has to be accounted for when the estimating the pool and fluxes. The mathematical models and different algorithms based on the in situ and experimental measurement of various parameters are then used to compute these values. There are wide geographical and seasonal variation in primary production in oceans. Most productive regions are upwelling regions associated with the current flowing along the west coast of the continents, especially namely the Canary currents in the northwest Africa, the Bengal current of southern Africa, the California current of California and Oregon, and the Humboldt current of Peru and Chile, and also the Somali current of in Western Indian Ocean, in Western Indian continent. Because of this, all these currents supports major fishery resources, and around the equator, the winds and Coriolis effects generated divergence current, resulting in an upwelling of dense, nutrient-rich water, which support the productivity of ocean. And there is also a large-scale upwelling in Southern Ocean, resulting from the replacement of northward flow of water driven by strong winds and Coriolis effects. Although productivity on the global basis is fairly constant across the year, it is highly seasonal in some oceanographic regions. The higher latitudes, the temperate regions, especially the North Atlantic, and characteristically show a spring bloom of phytoplanktons and subsequent zooplankton. The deep convective mixing occurs as a surface layer cooling during the cold, dark, windy winter, bring nutrients to surface layers and promoting a rapid increase in the photosynthesis process as light levels increases during the spring. The increased stratification and nutrient depletion leads to the reduction of productivity during summer, even though the light levels are at level their highest. During the autumn, the vertical mixing occurs again, and a small secondary peak of the production results. In tropical seas, like Indian areas, seasonal effects are much less pronounced, and except where there is a seasonal upwelling of nutrients, such as occurring in the Arabian Sea. The remote sensing can now also differentiate size class of phytoplanktons based on the models that allow us to estimate the photosynthetic rate relevant to the cell sizes, combined with the in situ measurements of abundance using a flow cytometry. This reveals the difference in geographical distribution and relative contribution of different groups of total production. The microphytoplanktons are abundant mainly at the mid to high latitudes, are patchily distributed, and nanophytoplanktons are moderately abundant globally, but concentration are higher at equatorial and mid to high latitudes, relatively low in subtropical gyres. But the phytoplanktons mainly dominate in oligotrophic gyres, and this is nutrient depleted areas in the ocean. The carbon is rarely a limiting factor in the productivity of oceans, since the bicarbonate is abundant in the seawater, but the nitrogen, phosphorus, silicon, trace metals, and some uh, trace organic compounds such as vitamin B can be limiting nutrient for primary production, thus affecting the primary productivity in ocean. The availability of iron is also a significant limiting factor in oligotrophic environments, has a significance. The penetration of water by the light of different wavelengths, limiting the photosynthesis in the upper 200 to 200 meter of clear ocean waters, considerably less in presence of suspended materials. Different phototrophic organisms are adapted to use the light of different wavelengths during different type of chlorophyll and auxiliary pigments, and this affects the distribution in the water column of different community of phytoplanktons. Very high iridescence can inhibit the photosynthetic process, especially due to the damage to the photosystem by ultraviolet light, and organism possess the photoprotective mechanisms to overcome these effects. And water column, the depth, water depth at which light intensity is just enough to balance the incorporation of the carbon dioxide by photosynthesis, and its loss due to the respiration is known as compensation depth. This vary according to the species composition of the phytoplankton, geographical region, and season, and also light penetration during the day and nutrient availability are the major factor deciding the compensation depth in the ocean. The classic food chain and microbial loop process occur in epipelagic region or the surface region of the ocean. The classic view of the marine trophic interactions are a simple pyramidal food chain, as we know, roughly summarized as the microalgae consumed by copepods, and these copepods consumed by fishes and whales. The development of microbial loop process concept has placed a much greater emphasis on the importance of dissolved organic matter in the water column, and the activity of bacteria, archaea, grazing protists, and fungi and virus. These are the microbes. They utilize this dissolved organic matter, and the trophic interaction multiple level, which microbial process occupy in the central stage, and these microbes utilize the dissolved organic matter available in the water column, and they retain the organic matter in the surface waters. This is significant especially in the oligotrophic environment. The microbial loop results in the retention of dissolved organic nutrients. About 50% of daily nutrient net primary production from the photosynthesis enters to ocean system as a dissolved organic matter, which supports the growth of heterotrophic microorganisms via microbial loop, resulting in greater retention of dissolved nutrients in the upper layers of the ocean. Some dissolved organic matter is formed from the diet, the direct extracellular release of the carbohydrates, amino acids, lipids, and organic acids from the phytoplankton cells as they grow. Under some circumstances, the phytoplankton cells can lose 5 to 10% of their cell mass a day. Greatest contribution of dissolved organic matter pool concern from the release of dimethyl sulfonopropionate DMSP from the microalgae. It has been suggested that no other single compound contribute as much as dissolved organic matter pool in the surface water. DMSP is also significant. This phytoplankton produced DMSP is also significant in climate regulation because this DMSP results in the formation of DMS, dimethyl sulfide, and cloud condensing nuclei formation. And this cloud condensing nuclei is significant in formation of clouds in our atmosphere. So these phytoplanktons are the they significantly contributing the formation of cloud formation in the oceanic environment. The dissolved organic matter and particular organic matter are also released during the grazing as a result of sloppy feeding by the zooplankton. When phytoplankton cells are broken apart by the mouthparts of the zooplankton, such as the copypods. However, by far most important source of the dissolved organic matter in the water column is lysis of phytoplankton and heterotrophic microbes by virus. Virus attack the phytoplankton cell and release the cellular material and form the dissolved organic matter formation. For example, up to 80% of the total photosynthetic production can be released as a dissolved organic matter during the collapse of phytoplankton blooms in the oceans. The biological pump transport fixed carbon to deep oceans and sediments. This is very important, and sequestration process in the ocean, controlling the carbon dioxide level in the atmosphere. And the gelatinous zooplankton species like the salps, larvaceans, and other mucus net feeding animals provide a mechanism for the export of particular organic carbon produced by the phytoplankton in the upper water column. These are the small pelagic barrel-shaped tunicates that pump large the volume of the seawater through their body and for movement of jet pulp, the propulsion, and trap the food particle in sticky mucus net that is rolled up and passed through their digestive tract. The salps package undigested material into large and dense fecal pellets of copypods and other rich in carbon that provide a route of rapid sequestration of particulate organic matter to deep oceans, deep sea sediment. Amplified by the vertical migration of salps to depth of 600 to 800 meter at night during the vertical migration. And the remaining of the dead salps also provide a large fraction of carbon reaching to our seafloor. The carbon export to primary production may change due to the ocean warming and acidification process. The alteration in the seawater chemistry as a result of ocean acidifications, which will have a large effect on the process of the calcification by the organisms such as coccolithophores. They are phytoplankton, planktonic organisms. Some studies based on the projection for future ocean acidification scenario indicate that such as the calcifying organisms could begin to experience a difficulty in maintaining their calcium carbonate skeleton as early as 2050. Especially in the polar oceans where the state of calcium carbonate saturation is very low. Some mesocausal studies of response of these coccolithophores like Emiliania huxley bloom to high carbon dioxide levels, stimulating end of the century conditions have showed a reduction in calcification rates when they were applied. But other evidence based on the analysis of geological sediment core suggests that the coccolithophores have gradually increased their overall calcification rate in response to past increase in the carbon dioxide levels. Some experiment studies have showed that some strains of Emiliania huxley increase both photosynthesis and calcification rate when high concentration level of the future predictions. The evidence for a possible negative feedback mechanism come from the observation of the cyanobacteria. It's a common blue-forming cyanobacteria in the oligotrophic environment, which is a trichodenmium, which increase in the rate of nitrogen fixation very markedly at high carbon dioxide levels. This could enhance the productivity of oligotrophic oceans that are currently limited by the nitrogen and increase the flux of carbon into the biological pump to the deep ocean. An alternative scenario is that warming, the freshening due to the melting of sea ice and changing the stratification of surface waters will lead to shift towards the dominance of small phototrophs whose consumption by grazing heterotrophic protists results in the retention of fixed carbon in surface layers rather than the transfer of higher trophic levels. The ingestion of the bacteria by the protists play a key role in the microbial loop in the ocean. The consumption of the bacteria by heterotrophs and microscopic small protists helps to explain the regulation of bacterial biomass at near constant levels. The bacteriophore constitutes a mechanism by which nutrients containing is a very small bacterial cells are made available to large planktonic organisms. Some metaphone zooplanktons like opipores can also consume the bacteria directly. Many pelagic bacteria and pico-eukaryotes associate with particles of marine snow rather than exist as a freely suspended forms. They may be consumed by a wide range of large metazoan zooplanktons and small fishes when they ingest these particles, although they be unable to feed directly on individual cells. An additional key component of microbial loop is the release of dissolved organic matter through the portistan grazing. The large amount of dissolved organic matter up to 25% of ingested prey carbon are ingested by the protists, and some of this is readily metabolized and enter to dissolved organic matter pool for further recycling by the heterotrophic organisms, whilst the remainder enters sink into the long-term refractory material, which is very significant in climate change scenario because this recalcitrant dissolved organic matter can be classified as semi-refractory dissolved organic matter or the ultra dissolved refractory dissolved organic matter, which is very significant because these are the microbes cannot be degrade this organic matter by biological process easily because this life is estimated as around the 4,000 to 6,000 years for degrade this recalcitrant dissolved organic matter. Therefore, this microbial carbon pump is very relevant in oceans. We have to study more on these aspects because this microbial lysis of the phytoplankton cells produce the recalcitrant dissolved organic matter rather than the labile dissolved organic matter. Labile dissolved organic matter can be utilized by the various trophic levels, but not by in the case of recalcitrant, it is sequestered for a long period. Then the viral shunt catalyze nutrient regeneration in the upper oceans. The viral lysis, as we told, this virus lysis is a significant contribution of formation of the recalcitrant dissolved organic matter in the water column. The viral lysis of the primary producer and the heterotrophic organisms act as a shortcut that disrupt the flow of nutrient into higher trophic levels. So it is called viral shunt. The viral lysis leads to the release of cell content, much of which is entered to a dissolved organic matter pool, is readily recycled by the heterotrophs. However, the cell fragments and high molecular weight polymer are more recalcitrant to break down by bacteria and other biological components, such as the phag lysis of the bacteria leads to release of cell envelope fragments containing embedded bacterial outer membrane proteins, which is relatively resistant to proteolite degradation process. Some components such as the cell wall polymers of the algal cells lysed by viruses are also very refractory in nature. The quantity and the dynamics of this less labile component resulting from the viral lysis are not yet fully known. We need to study much more on these aspects to understand the carbon sequestration process in the ocean. The another process is the eutrophication of the coastal water, stimulate the microbial activity and improve the productivity, and it will result in the negative impact on our environment. The increased nutrient loading stimulate the phytoplankton growth beyond the point at which it is controlled by zooplankton grazing and or by the virus infection. Active microbial loop process convert the excess of primary production, but this too much of overwhelmed and large amount of decaying detritus and particles of the organic materials will sink towards the deep sea floor. These sinking particles gets to bacterial decomposition leads to the heavy demand for the oxygen, the water column, and deep sediment, and results in the mass mortality of benthic animals and fishes. The number of such dead zones in shallow coastal waters are approximately doubled since the 1960s. As we know, the increased oxygen minimums on Arabian Sea and northern Indian Ocean regions are a result of this process. Even slight reduced oxygen levels may disrupt the food chain and cause stress-induced diseases in economically important fish and shellfishes, as well as these local effects. The expansion of dead zones may have a serious consequence for our climate change. The heterotrophic denitrification microbes leads to the production of greenhouse gas such as the nitrous oxide through this process, and elevated atmospheric concentration of this nitrous oxide and other greenhouse gas could further exacerbate the effect of global warming and contribute to ozone depletion and causing the increased exposure to harmful ultraviolet radiation to our Earth. In conclusion, in the increased use of the autonomous sampling and monitoring devices and development of large-scale mesocosm experiments for the study will be very viable and is very important. And this is an area of intense research happening, and the coordinated methodological approach are leading to further insight into the possible future trends in this process, and increased efforts are now need to gain further understanding on integration of these systems and how they will respond to changes in physical, chemical, and hydrographical properties of the oceans as a result of the human accelerated or human induced anthropogenic activities in our ocean. So we have conclude here, and I'm thankful to organizing committee for the opportunity given for me, especially Dr. Ansari for this opportunity. Thank you all.
Thank you, Dr. Jayachandran. Your wonderful talk, and then we happy with you with us. So now the session is open for discussion. If anyone having the questions or any problems you want to solve with him, please get the suggestion from you. Participants, if you have any doubt regarding Dr. Jayachandran's talk, please interact with him, because the interaction will make more ideas what we are getting.
Anyway, sorry, initially I could not share the screen and some
No issue,
problems.
no issue. Yeah. So this is the issue we always having in the virtual platform. So anyhow, we are just adapting.
Okay,
Maybe in
okay.
future we will be get in touch. I think hopefully there is no question. So I feel Jayachandran talk was very interesting, as well as people can get understood. That is the reason there is no much question about it. There is a two-way possibilities. One might be understandable, otherwise people are not understanding. Okay. If you have
Okay,
any
sir.
questions or doubt, we will share the contact information of Dr. Jayachandran. Uh,
Okay.
you can get the direct queries towards the mail, and then you will get the suggestion and then ideas from Dr. Jayachandran. So we
Okay.
hope we will end this session. Uh,
Okay. Okay.
we will end this talk in this session, in the first half. So we will give 10 minutes break to join exactly 11:30 for the same link, using the same link for the next speaker, Dr. Mufiz, because he's in almost in 6:30 from his country. So I'm just getting contact with him. Let's see the 10 minutes break. Please, all the participants will join with us in exactly 11:30. Thank you.
Thank you.
இது அப்படியே இருக்கும். இங்கிலீஷ் இங்கிலீஷ் பண்ணியிருக்கோம். இப்போ வந்து என்னன்னா இப்போ ரெக்கார்ட்
Hello, good morning from Dhaka. Can you hear me?
Yes, sir. I am getting your voice. I hope. Welcome to our session.
Thank you very much.
Okay.
It's good to see you. Thank you.
Yeah. Thank you, Mufis. We will be very long meeting. Okay. 1 or 2 minutes. We will start the session.
Sure, sure. I'll be waiting for that.
Yeah.
So if I wanna share my screen, is it possible? Oh yeah, I can see it. The present now.
சரி அஷ்னா
You can
டென்டல்
share.
ஃபோன் வந்துச்சு சார் ஒரு நம்பர் கேட்டு இஸ்மாயிலா அனுப்பிட்டேன் சார்
Oh, I need permission from you to share my screen of my presentation.
Yeah. We will be. I will make it that no issue.
Yeah.
Okay.
Yeah, good morning once again. It's audible?
Good morning.
Now it's time to hear the words of keynote speaker on the second speaker of the technical session 1. And now I request Dr. Tamim Ansari to introduce the keynote speaker. Over to Dr. Tamim.
Okay. Thank you, Dr. Feroz Khan, to give the good opportunity for giving Dr. Mofis to introduce. So our the keynote speaker, Mohammad Mufizur Rahman, currently working in TH Cologne University of Applied Sciences, German. Basically, he's from Bangladesh. He's schooling and then bachelor degree he completed in Bangladesh, particularly in environmental science, bachelor of science in environmental science, in life science school, Khulna University, Bangladesh. And he got a prestigious MSc fellowship from European Union, and then he joined his MSc Marine Biodiversity and Conservation in the great University of Ghent, Belgium. After that, he also have lot of diplomas, PG diploma in even he studied in India, IIT Madras, in Sustainable Development Program in IIT Madras, and then he have the very good knowledge on transdisciplinary environmental scientist he known to be as. So he used different mechanism to find out the human relationship with environmental conditions. So we have the very good person to get the day-to-day information what we are making on the climate change and the adaptation. And then he is the receiver of different fellowships and then the grant, and then he also the prestigious grant he got Alexander van Humboldt fellowship climate for the climate science. So we used to call movies. He's working in everywhere in the world. What I know from him from last couple of years. So he also published in various papers in journals, and then he wrote different type of environmental related reports for different countries, particularly on German. And then he also presented the different type of conferences, particularly in Netherlands, Scotland, and then German. Even in India, the 2019 we have a meet. So he gave the very good presentation over there. So now I invite Mr. Mufiz Rahman to give his findings and then his expertise to our beloved participants through this our conference. So I welcome Mufiz to start your presentation. Now the stage is
Thank
yours.
you.
Thank you.
Thank you, Dr. Ansari. The problem is that I cannot share my screen. It says it needs permission. Is it possible?
You just start to you just start to share your slide. I will just
Okay.
give you permission.
So thank you,
I
Dr.
hope.
Ansari. Dr. Ansari is a good friend of mine. I met him first time in Chennai long ago. Then I met him in Calcutta in ISER. So it's good to see that he's one of the organizers of the conference, and I'm glad that I'm invited. Uh, so today, even though the conference is about zoology, and I see very many of you are working in the department of zoology, uh, but my talk will be quite broad. My research is focusing on river deltas, and as you all know that deltas are very fragile ecosystem. Uh, so deltas are usually, you can find them on the river mouth. For example, we have one of the greatest delta, one of the largest and fertile and volatile deltas in the world called Ganges Brahmaputra Meghna Delta, which is located in on the mouth of Ganges Brahmaputra Meghna.